Let's start with question number one. Describe the induced fit model of enzyme action and how an enzyme act as a catalyst. Good. So why it's a catalyst? So how do we define a catalyst? That speed up the reaction without being used up by lowering the activation energy. So we have mentioned this point. And what sorry, about the induced fit model, you have told it very right, that substrate bind to the active site, and then the shape is slightly changed to fix in or to fit in the substrate that should be complementary, and the bonds are going to form temporarily, and finally the substrate is converted into like product. So the next one, scientists investigated the action of enzyme ATP synthase. They made reaction mixture containing ATP synthase, buffer, and a substrate. So tick one box to show what other substrate the scientist must add to the reaction to produce ATP. What is required for making ATP? It's related to respiration as well. What? How is ATP formed? By putting the phosphate in ADP. So we say that in yes, ADP plus the inorganic phosphate gives ATP. So we need adenosine diphosphate. Let's move to the next question. So the next question states, so there is a graph. Let's move to the graph. So there is a graph that scientists, wait, okay, the scientists investigated the effect of concentration of inorganic phosphate on the activity of enzyme synthase. After 2 minutes, they stopped each reaction and then measured the concentration of ATP. So this is how the result looks like. So how would you suggest and explain a procedure that scientists could have used to stop each reaction? How the reaction is stopped? For example, if some so first of all, they increase the temperature to denature enzyme, increase the temperature to denature. Okay, what else can be done? They are using buffer as well, so they can change the pH by adding no, buffer is already present. Look over here, buffer is given acid, the strong acid, because the weak acid is not going to affect the buffer solution. The strong acid, okay. Okay, two reasons are enough. What else can be done? We can add one more thing, the temperature, the pH, and inhibitor. Yes, we can add inhibitors to stop the reaction. Then our substrate is not going to bind that enzyme. Okay, the last one part, explain the change in ATP concentration with increasing inorganic phosphate concentration. Now you have to look at the graph and explain the change. This graph, explain it. Like what is happening while we when we increase the concentration of inorganic phosphate. So we are increasing the concentration of ATP produced. There is more ATP produced, or we can say more enzyme substrate complexes formed. So we have told them the start. This is the basic thing. Now the optimum point. What is the optimum point? That at 40 millimoles per cubic decimeter, 40 millimole per cubic decimeter, the concentration is now constant. Why it becomes constant? Because all the active sites is occupied active sites. Great. Or we can also mention that it would become, the concentration would become limiting factor. Okay, let's move to the next question. Explain the disadvantages for larger animals for having a specialized system that facilitates oxygen uptake. How it's going to benefit us? Exactly. They would have a bigger surface area to volume ratio. Okay, bigger. You have given me 1 point. Bigger, smaller area, surface area to volume ratio. What else can be the reason? You have told me correctly that the bigger animals. Okay, so they're faster diffusion. Okay, faster diffusion. So we say the larger organisms have smaller, not bigger. I've written for the smaller animals. The larger animals have smaller, and we need the smaller surface area to volume ratio to facilitate diffusion, or the faster diffusion. Okay, again, the next, there is a diagram given, and there are two models given. Let's discuss those models, and let's see what are these models. So we have two models. One is oxygen uptake through the system developed to the outside of the body, for example, fish lungs, and one is the related to human lungs, that is alveoli. Suggest how the environmental conditions have resulted in adaptation of a system using model A and model B. Like why their structure is different according to the adaptation of environment, the environment in which they are living, and why the structure becomes different. First of all, what is the like habitat of fish? Water. So does water have the higher oxygen concentration than air, or air has the higher concentration than water? So the concentration of oxygen. Our main goal is to get the oxygen, right? So let's say water has less oxygen concentration. Okay, what would be the effect on this one? How would it affect the adaptation? Like their lungs would be developed. Wait, they have larger surface area because they're outside the body, we can say. It's written it's outside the body, so there would be larger surface area to absorb more oxygen from water. Okay, what else can be done? Else can be written? So these are 2 points. What would be the third point? First thing we have mentioned water, the concentration. Second thing we have mentioned the surface area. Now again we have mentioned, for example, we have mentioned the surface area, and then we have said that there would be faster diffusion when the distance is, sorry, when the distance is shorter. Okay, distance is shorter, so there would be better diffusion. Okay, next question. Again, uh, diagram is given. Let's move to the diagram. So there is a human exchange system, and then there is a fish exchange system. So the key has been given: what is I, what is E, what is A, and what is V. So A and V is straight away artery and vein. Okay, E is representing the exit, and I is representing the entry. Okay, so a student studied figure 3 and conclude that fish gas exchange system is more efficient than human gas exchange system. Why do you think so? So there is something overlapping of something in fish, right? Okay, correct. V has less. Wait. Are you saying V has less oxygen than E? Okay, in humans. Okay, okay. V then E. Okay, this is the one difference. What other thing can be written? Difference in concentration of oxygen. Re- That makes it easier to remove oxygen, or that it makes, sorry, not remove. Um, yes, remove. It would be remove. Explain how the counter current principle allows efficient oxygen uptake in fish gas exchange system. Now it's counter current. If one thing is like moving in like clockwise direction, the other would be going in anti-clockwise direction. In opposite direction, yes. So blood in opposite direction. So a concentration gradient is maintained. Is maintained where? Lamella. Okay, good. So the next question. Again, regard. Okay. Calculate how many times. Wait, let me first of all copy it. Okay. Table 1 shows the feature of two mammals and their mean body mass is given, and according to that the mean lung volume is given. Okay, so what they've said calculate how many times the lung volume per unit, okay, of body mass of bat is greater than that of shrew. Yes. Okay, and what would be your answer? Can you calculate the answer for me? Okay, 130. 130, okay. And in shrew, it's 30, correct. Okay. So give the answer in appropriate. Okay. So suggest one, give one suggestion to explain the difference. No, okay. If you want to tell ratio, first of all, how many times it would be greater? The ratio of greatness. It would be 130, you have told, and it is 30. Okay. What is their ratio? Yes, divide them. Three times more. Okay. So if we have to write answer in two significant figures, what it would be 4.3. Okay, 4.33. Okay. Now what is the explanation, or what is the one explanation or the one suggestion to explain this difference? Again, we are talking about the same thing from the start. Okay. Provide more oxygen for respiration. Correct. Okay. Why do you think the bats need more oxygen? Because they need more oxygen in flight. They're using more energy in flying than shrews. Than shrews. Though this reason is not asked, I'm just asking that why do they need more oxygen as compared to shrew? Why there is adaptation? Okay, let's move to the next question. Describe how one amino acid is added to polypeptide that is being formed at a ribosome during translation. So it's related to translation, how the amino acids are added during. Correct. So first of all, you have mentioned tRNA. It brings the amino acid to that polypeptide chain. Uh, then you have mentioned what? Codons or anticodons? Okay. What have you said? The anticodons on tRNA binds to the codon by that you have mentioned by condensation or the peptide linkage. Amino acid joined together by condensation, also known as peptide linkage or peptide bond. Okay, this is all. So let's move to the next one. Again, a table. Oh, okay. There are amino acids given and a table is given. Like, which sequence can we use? Sorry. Wait. Let me copy paste because we need to explain it according to this. Okay. A key has been given. So, crystalline is a structural protein found in human eye. A inherited disease that leads to the blindness is caused by the change in the property of crystalline. The replacement of amino acid arginine over the amino acid glycine causes such changes. Use the table that how why this amino acid replacement changes the property of crystalline. Now we have to use the sequence from this table. Okay. Okay. How? How? Arginine to glycine. They have told that it has been like what changes the structure, like which bonds. Like look at this one. Which bonds are going to be changed? The lighter one are hydrogen bond. The darker one are. Wait. Uh, yes. So instead of arginine, we say here we have arginine, which is, so what is present instead of arginine? Glycine. So where is glycine? Let's go to, okay, glycine. So this one is glycine. So this bond is going to be affected because glycine is there now. So okay, if glycine is there, then hydrogen bond instead of, sorry, yes, hydrogen bond instead of ionic bond, and it will change what structure? Because they have talked about the structure. Sorry, the tertiary structure. The tertiary structure. So we have to write these 2 points in detail. Okay, let's move to the next point. Again, a table is given. We need to complete the table, and I'm pretty sure you that you will be able to pull it really easily. Let's go to this question. The amino acid replacement of ARG with glycine is caused by a single base substitution mutation in DNA. The non-mutant DNA triplet is TCC. Given what is given, the mRNA codon complementary to the non-mutant DNA triplet, the mutated mRNA codon that causes the change from arginine to this, and the DNA triplet that complementary to this mutated one. Okay, let's find what are the codes present in the table. Here is a table. If it's is arginine would be AAG, and glycine would be like UGG, it can be CGG, it can be AGG, it can be GGG. So this, I guess, yeah, mutated one would be, okay, let's say it would be GGG. And what would be the DNA? Yes, C or complementary to this one. Okay, what would be the mRNA codon if I look at this? Wait, wait. If we discuss these two, G, it would be A, G, and G. So it would be G, G, and G that we have mentioned. This is how we make codes, right? First take from this one, then from this one, whatever, and then from the last one. Okay. So it would be A, G, and G. Why not A, G, A? Why not AGA? Because both are like coding for ARG, AGA and AGG. AGA, AGG. So we're talking about the non-mutant DNA. That's why we have selected AGG. Okay.